So today through this video I will be discussing about the Mendelian dihybrid cross. In my previous video actually I have discussed there about the Mendelian monohybrid cross, and now in this video I will be discussing about the Mendelian dihybrid cross. Okay, so dihybrid cross means the cross between two pair of contrasting characters. In the previous one, in the monohybrid cross, I have taken one pair of contrasting character, and if you observe that video, in that video I have taken one character height, and in that height two contrasting character tall and dwarf it was taken. But now in this dihybrid cross I have to take two pair of contrasting character. That means if I take height, that is one character, so I have to take another character. I have to observe another character. Suppose I am taking here seed shape. Okay, seed shape. Suppose I am taking here. So let's begin. So I am writing here Mendelian dihybrid cross. So I am taking here Mendelian dihybrid experiment. Okay, and I am taking two pair of contrasting character. So I am taking suppose tall pea plant with red. Suppose I am taking the shape of the seed, tall pea plant with round seed. So I am taking the height and I am also taking the shape of the seed. Okay, so that's why I have written tall, which is indicating the height of the pea plant, and round, which is indicating the shape of the seed. Okay, which actually Mendel has given in his experiment. So tall and round, and it is mainly making cross with the dwarf and wrinkled seed. So here the tall and round, tall, this is for the height, this is the dominant trait of the height, and round, this is the dominant trait for the seed shape. Dwarf, this is the recessive trait for the height, and wrinkled, this is the recessive trait for the seed shape. Okay, and as we all know, for the dominant trait we are just taking the first alphabet. Okay, and we need to write it in a capital letter. So suppose tall, here it is the dominant trait, so we have to take capital T. Okay, and round, this is also a dominant trait for seed shape, so we are taking capital R. Okay, so capital T and capital R, capital R. Now the question, why I have written two times capital T, and for round I have taken two times capital R. Why? So if we see the chromosome, and in my previous video I already told there that pea plant, this is the diploid organism. That means two set of chromosomes are present, and in a particular locus the alleles are present, and these are mainly controlling same kind of characters. Okay, so suppose here this particular portion, it is mainly regulating the height of the pea plant, and suppose this particular locus, it is mainly for the genes which are controlling the shape of the seed. Okay, so here capital T, this one I have written, capital R, this one I have written. That means here also in the same way I can place here diploid. Okay, two set of chromosome, two set of homologous chromosome. In this locus the genes are present which are indicating, which are mainly regulating the dwarfness, dwarf trait. Okay, and here in this position, suppose the genes are present, but these genes are not actually regulating the height, but they are controlling the seed shape. Okay, the recessive trait, they are mainly controlling smaller. Okay, so I have to write here small t, smaller. Okay, so this way I have to write. So I am just erasing this portion. So capital T, capital R, and here small t, smaller. Okay. Now I have to segregate the gametes. So if we observe the previous feature which I have drawn here, capital T, capital R. If we observe, and as you all know, in the gamete, a single set of chromosomes are mainly transferring. That means Suppose this particular portion is mainly transferring to the gamete. That means we are getting capital T, capital R. Okay, it is indicating the gamete. Here also in the same way, small t and small r. Suppose this particular portion of the chromosome, okay, the single set of chromosome is mainly transporting to the gametic part. Okay, so here we can write small t, small r. So here these portions, these are mainly indicating the gametes. So these are indicating the gametes. So I am writing here gametes. These are indicating the gametes. Okay, so this information which I have given, this information, it is mainly for parental generation. Okay, these are the phenotype of the parental generation, and these are mainly the genotype of the parental generation. And I have segregated the gametes. Okay, from capital T, capital R, the gamete which will be coming here, capital T, capital R. Okay, out of two, one is coming, one T is coming here, and out of two R, one R is mainly coming here, because this is gamete. Here also same way, out of this small t, one small t is mainly coming in the gamete, and smaller, two small r is there, and one small r is mainly coming or transferring to the gamete. Okay, because through the meiosis, half number of chromosomes are mainly transporting or transferring. Okay, now these gametes are mainly formed from these parents. Okay, now we have to make a cross here. So just to the cross, what we'll get? Okay, what I have to draw this part again. So we are getting capital T, capital R from this part, and we are getting small t, small r from this part. So what we will get finally? We will get capital T, capital R, small r. Okay, capital T, capital R, small r. So this one we finally get. So you can write here capital T, you can write here capital T, capital R, small r. So this is the offspring which will be obtained in F1 generation. This is the offspring which will be obtained in F1 generation. This is the genotype of F1 generation. So what will be the phenotype? The phenotype is, as capital T is there and T is the dominant, so tall, and R is the dominant, which is indicating round, which means round. That means in F1 generation, we mainly obtained tall pea plant with round seed. Okay, this is about the F1 generation. This is the genotype of F1 generation, and tall round, this is the phenotype of F1 generation. Now we have to do the selfing. Selfing means the offspring which mainly obtained here, we have to make cross in between the same offspring in selfing. So just do the selfing here. Selfing. In selfing, we are just making cross in between the offspring of F1. So F1 cross F1 means F1 offspring is making crossing with the F1. Okay. So what we will get actually? Capital T, capital R, small r. So in the F1, we are just obtaining this kind of genotype. And okay. And in the F1, so they are just making cross in between. So again I have to write capital T, capital R, small r. Okay, suppose 4 offsprings are obtained in F1 generation, and out of these 4, 2 offsprings are again doing the selfing. Okay, so that's why I have written capital T, capital R, small r, cross capital T, capital R, small r, which we mainly obtained in F1. Now I have to segregate the gametes. So segregate the gamete, and in this gametic segregation, just see what kind of possible combination of the gametes we can obtain in this type. So suppose I am getting here capital T, capital R, small r. Okay, so what is the possible combination of the gametes which we mainly obtain from them? So just see. This capital T, it may come with this capital R, so TR, it may come as gamete, or This capital T is mainly come with smaller, that means capital T smaller. This is the another possibility. Or small t is coming with capital R, capital small t capital R. This is the another possibility. Or small t, it is coming with the smaller, that means small t smaller. This is the fourth possibility. Okay, so these are the possible combination of the gametes which we can obtain from F1 generation. Okay, so capital T capital R, capital T smaller, small t capital R, or small t smaller. With one t, another R is coming. Okay, see, T R, T R, T R, T R. Okay, that means from this type also same kind of gametes will be obtained. Capital T capital R, or capital T small r, or small t capital R, or small t smaller. Same only. Okay, so these are the possible combination of the gametes obtained from this, and these are the possible combination of the gametes obtained from this. Okay, so now we have to place them in a Punnett square. So place them in a Punnett square. What we obtained there? We have to make a table first. Okay, so this side here I will place the female gametes, and here I will place the male gametes. And I obtained actually capital T capital R, capital T small r, small t capital R, or small t smaller, which I obtained as gamete. Okay, so here also same, capital T capital R, capital T small r, small t capital R, and small t smaller. Okay, so I placed the gametes. Now make it cross. So capital T capital R, capital T capital R, capital T capital T, capital R, capital R. Okay, this we have to write. This one and this part, capital T, capital R, smaller. Okay, now this part and this part, capital T, small t, capital R, capital R. Okay, now this one and this one, capital T, small t, capital R, smaller. Okay, now this one, this gamete and this gamete, capital T, capital R, smaller. Then this one and this one, capital T, smaller, smaller. This one and this one, capital T, small t, capital R, smaller. Then this one and this one, capital T, smaller, smaller. Okay. Then this one and this one, capital T, small t, capital R, capital R. This part and this part, capital T, small t, capital R, smaller. Then this part and this part, small t, capital R, capital R. Then these two part, small t, capital R, smaller. Okay. Then this part and this part, capital T, small t, capital R, smaller. Then this part and this part, capital T, smaller, smaller. Then this part and this part, small t, capital R, smaller. Okay. Then last part, small t, smaller, smaller. So these are the offspring which we mainly obtained in F2 generation. So these are all indicating the genotype. Now in bracket below I have to write the phenotype also. So capital T, capital R, capital T, capital R, capital R. That means tall and round. Tall and round. Here is also tall and round. That means tall pea plant with round seed. Then tall, again capital T, small t, that means tall and round. So already I have written the genotype, now I am placing the phenotype below this. Here capital T, small t, that means tall and capital R small r means round. Okay, round. Here capital T that means tall, and capital R small r means round. Okay. Here this part capital T means tall, and small r means wrinkled. Okay, wrinkled. And then capital T small t that means tall, and capital R small r means round. Okay. Then this part capital T small t means tall, and small r means wrinkled. Okay. This part capital T small t means tall, and capital R means round. This part capital T small t means tall, and capital R small r means round. Then small t means dwarf, and capital R means round. Okay. Then small t means again dwarf, and capital R small r means round. Then this part capital T small t means tall, and capital R small r means round. Okay. This part capital T small t means tall, and small r means wrinkled. Then small t means dwarf, and capital R small r means round. No, because capital R is the dominant one. And small t means dwarf, and small r means wrinkled. Okay, because small r is the recessive form. So these are the genotype which I have written below the these are the phenotype which I have written below the genotype. Now if we observe the phenotypic ratio of F2 generation, now what will be the phenotypic ratio? So I am writing here the phenotypic ratio of F2. What will be the phenotypic ratio of F2? So what type of character we mainly obtained here? If you see, we got tall, round. So tall, round is 2. Then similarly you just see, if new part you will get tall, wrinkled. We also get tall, wrinkled is 2. Then similarly go there, go see, check. Okay, dwarf, round, that's also new. Dwarf, round. Then again you go thoroughly, and then the another type I got is the dwarf and wrinkled. So tall, round. How many tall, round plants actually I obtained in F2? So you have to count tall, round. 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, so 9. Then tall, wrinkled. How many tall, wrinkled I actually got there? Tall, wrinkled. 1, 2, 3. So 3. Dwarf, round. How many dwarf, round I will get actually? 1, 2, 3. 3. And how many dwarf, wrinkled? Only 1. So 1. 9:3:3:1. So 9:3:3:1. This is the phenotypic ratio of F2 generation, which we mainly got in Mendelian dihybrid process. And if we observe the genotypic ratio, it is very complex. But I am telling you what will be the genotypic ratio we get from there. That means we have to arrange these alphabets there, which is indicating the genotype. Okay, and just we have to observe their frequency and how many we will get there. So the genotypic ratio which we will be obtaining F2, it will be 1:2:2:2:4:1:2:1:2:1. Okay, this is the genotypic ratio which will be obtained in F2 generation. You can check it if you just place them these genotypes. Okay, and just count their frequency. Okay, number of repetition of these alphabets, and then you will easily get the genotypic ratio which I told. Okay, so this is about the Mendelian dihybrid cross. So thank you for watching this video.